Hi, everyone. Welcome back to Spatial Omics Seminar Series 7. And after a couple of months, I think we're excited to get started again and to share the recent updates with the community. And we're also happy to have today Professor Sten Linnarsson. And briefly, I'll be introducing him. And Professor Sten Linnarsson took his PhD in 2001, studying neurotrophic factors regulating neuronal survival, growth, and plasticity. Instead of a postdoc, he founded a company to develop methods for gene expression analysis and single molecule DNA sequencing. In 2007, he was appointed assistant professor, and in 2015, professor of molecular systems biology at Karolinska Institute. And he was awarded the 2015 Eric Fernström Prize for his work in single cell biology, and is a member of the European Molecular Biology Organization, for short EMBO, and the organizing committee of the Human Cell Atlas Initiative and the Nobel Assembly at Karolinska Institute. And since 2007, Linnarsson has pursued single cell biology with the ultimate aim to discover the complete lineage tree of the developing human nervous system. To achieve this goal, his group pioneered single-cell RNA sequencing, RNA single molecule FISH, and advanced computational methods. And beyond, also earlier contributions included the integration of unique molecular identifiers in the single-cell RNA sequencing field, and recent advances involved RNA velocity, a transformative concept in the RNA sequencing field. And today we're going to learn about actually new technology that Professor Linnarsson developed. With this, without further ado, I'll turn over to Professor Linnarsson.
Thank you. Thanks so much for that introduction. Let me share my slides. Right. So we're going to talk about fish today, and specifically electric fish. So I'm happy to very grateful to be invited to your seminar series here, and happy to present our new method that uses electricity to improve methods for RNA fish. So let's see if I can flip my slides here. Right. So the method I'm going to present is called enhanced electric fish, or eel fish for short. And this was really the brainchild of a brilliant student of mine, Lars Borm, who I think may be online here, who came up with this excellent idea of using electrophoresis to transfer RNA out of tissue sections and onto a glass slide. As well as Simone Codiluppi, who has been a senior staff scientist in my lab for several years, many years, and worked with our many efforts to develop and improve methods for RNA fish, RNA in situ hybridization. Right, so before I go on, I have to disclose that the method I'm going to present has been patented, and we licensed this to Rebus Biosystems. So whatever I say, you have to just keep that in mind, that I have this disclosure. So a few years back, we developed a method that we called Awesomefish. And this was really a relatively straightforward cyclic fluorescent in situ hybridization method, where we optimized the chemistry and the instrumentation for repeated hybridization of single molecule probes for specific mRNAs. So the image you see here, it shows mainly the somatosensory cortex of the mouse. And you see the legend here shows which genes we detected here. And this was done by repeatedly hybridizing and detecting a small number of genes, one set at a time, one after the other. So no barcoding or anything like that. But it's kind of the gold standard single molecule fish, which was, I think, first developed by Arjun Raj, really has the power to detect individual molecules with extremely high sensitivity in situ. So you can get these beautiful images of individual RNA molecules in the tissue, and you really here make out the cells very nicely. Now, the problem with Awesomefish was mainly the imaging speed. So it took us about 3.5 hours to image a square millimeter. The somatosensory cortex that you see here on the left, it's a few square millimeters. And so each imaging round took many hours, and then it's a cyclic process. So you have to repeat this for many cycles. In total, it took us about 2 weeks to acquire the image you see here on the left. And to be honest, it was never really robust because, you know, if you have an instrument running nonstop for 2 weeks, there are many things that can go wrong. So Eel, which I'm going to talk to you about today, mainly improves the imaging speed. So the main purpose of Eel is to increase or scale Awesomefish to larger numbers of, to larger areas. And also as a side effect of the way we decode the images to larger numbers of genes. So if you compare Awesomefish on the left and Eel on the right, you can see that the speed up is tremendous. We go from 3.5 hours for a square millimeter to 62 seconds for a square millimeter. And I'll tell you in a minute why this is. But the great advantage of Eel then is that it allows us to go to centimeter scale specimens. Like here, it's a full coronal section of the mouse. And the images that you see here on the right, this was acquired in just 2 days of imaging. So it's a much larger area, it's many more genes, and it takes only 2 days instead of 2 weeks. Right, so how do we do this? So it was the insight of Lars to, wait, maybe I should go back here. So the issue in imaging here is, or the bottleneck in setting the speed and the time it takes to perform an experiment like this is really the imaging. And the imaging, of course, is the imaging speed is a product of the dimensions that you try to image, the total area, but also of the vertical imaging that you have to do. So when you image tissue like this with high power optics necessary to image single molecules, you have to image a stack, a Z stack. So on the left here, I think it's more than 30 images deep. But this really doesn't provide much information because the Z resolution of epifluorescence microscopes is not very good. And the first step of analysis anyway is to make a max projection of the Z stack. So we're acquiring this huge amount of images in X, Y, and Z. But then the first thing we do once we get these images is that we collapse the Z dimension into a single flattened image. Now, Lars thought about this and basically realized that if we could perform this collapsing of the Z dimension physically instead of in silico, then we would only have to image a single Z plane, and that would greatly speed up the imaging, potentially, you know, 10-fold or 30-fold or more. And so the approach he came up with was to deposit the tissue section on a glass slide that would have a surface chemistry designed to capture mRNAs and would be transparent for imaging, but at the same time would be conductive. So this is a glass slide coated with indium tin oxide. It's a thin layer of metal oxide that's conductive but transparent. And now what we can do is we can apply a voltage gradient across the tissue and basically use electrophoresis to drive the RNA molecules onto the surface. So we apply a voltage potential across the, in this case it's a flow cell, it's like a sandwich, and we make the capture slide the positive pole of this gradient, and so the RNA molecules migrate towards the surface, and then they're captured by, in this case, oligo-T primers here, oligo-T oligos. Once the RNA has been captured on the surface, then we can digest away the tissue. So we end up with a glass slide that has more or less only the mRNA molecules captured on it, and all the tissue is gone. So in this way, it's a bit like clearing, but it's like a supercharged version of clearing where we actually physically get rid of the whole tissue section. Now, this gives us a couple of advantages. First of all, the RNA molecules are now in a single plane. So we no longer need to image in the Z dimension. We can image only in X and Y. And it reduces background fluorescence from the tissue section. So it definitely greatly increases the signal over background ratio. And we took advantage of this increase in the quality of the images by implementing a barcoding scheme. So whereas in our previous method, we read out genes one by one in each color channel, now we use a barcoding scheme. So we hybridize onto each mRNA molecule, we hybridize primary encoding probes that are depicted here as sort of staples here. So there's a number of these hybridized to each target mRNA molecule. And then we have secondary readout probes that read out these encoding probes. And so each gene is encoded by a particular combination of positive signals across 16 cycles of hybridization. And with the encoding scheme that we use, this allows us to encode up to 448 genes in 16 cycles of hybridization, where each gene is 16 cycles with 6 bits that are on, so visible signal, and 10 bits that are off, so background signal or no signal. So this allows us to scale up the number of genes that we can detect. The electrophoresis setup is very simple. Here on the left, you see a rendering of it, but this is really how it works. So you have 2 slides that are positioned as a sandwich, as I mentioned. Both of them are conductive, and we attach these conductors to the slides, and then we apply voltage. We don't actually use a battery, but we do use a low voltage, like 1.5 volts. We use a more controlled power source, but in essence, this is all you need to do the transfer. And then Lars implemented this in the form of a fully automated microscope system that has, it has the fluorescent readout probes, it has the buffers and so on, and there are valves and syringe pumps to, in a controlled way, repeatedly deliver readout probes, perform the imaging, and then strip and wash the previous readout probe. So we have now 3 of these systems running in the lab, and the systems are really workhorses. They work, we can perform these experiments repeatedly and in sequence to generate multiple data sets in quick succession. So this is a much more robust method than what we previously worked with. All right, so now we come to the most interesting experiment in this part, in this project. Lars got worried that if we're performing electrophoresis and we're using 1.5 volts, then we actually might be splitting water here, because the electrolysis sort of threshold for water is 1.23 volts. If you apply more than 1.23 volts to water, you split it into hydrogen and oxygen. So it might be that if you do this, if you apply, as we do, 1.5 volts to the slide, you might actually be not just electrophoresing the RNA towards the anode, but you might also be splitting water at the anode. And if you do, then you can see the reaction scheme here. You will generate some oxygen, but would also generate some protons. And so this might then cause a drop in pH at the surface. And if pH drops at the surface, then RNA will no longer hybridize because the melting point will be strongly skewed by the acidic conditions. So maybe this is a concern that might make this whole method impossible, basically. So this prompted Lars to come up with a beautiful experiment to measure pH at the surface. It turns out that there are bacteria that you can buy, so E. coli basically, that you can buy that are dead, but I think more or less dead, but they contain pH sensitive dye or reporter. So these bacteria, they will shift their fluorescence as a function of pH. So what we did was we deposited these bacteria on the surface, and then we deposited the tissue on top. So now you have these pH sensors that are really micron-sized pH sensors between the tissue and the surface. So really exactly where we want to measure the pH. If we now apply a voltage gradient across here, we can by looking at the fluorescence of these doped E. coli, we can measure the pH exactly at the capture surface or within a micron of it. So this is what they look like in B here. And in C, we generated a calibration curve. So you can see that at neutral pH, the fluorescence is low. And then as the buffer gets more acidic, then the fluorescence intensity increases. So this allows you to measure pH at the surface. Now, luckily, it turns out that in the conditions that we're using for eel, which is 1.5 volts, basically nothing happens. So the fluorescence stays flat. There's a slight increase here maybe when you turn on the electricity, but it's really nothing to worry about. The change is, you can see the scale is the same as we go from 0 to 2000 fluorescence units. So it stays flat here means that it's really above 7, the pH. Now, just to verify that the measurement system works, we also applied 6 volts. And if you do that, then you can see actually a very nice, clear, distinct drop in pH when you apply the voltage potential. So you have here just a few second pulse of 6 volts of voltage, and you can see the pH stays flat, and then it drops, and then it goes back. And here you can see also see the sorry, the electricity is on here. This is the bar that shows when the electricity is on. So when you switch off the electricity, the pH goes back again as the buffer diffuses in under the tissue. In just a few seconds, it's back to 7. And these images here show how the measurements are actually performed. You can see these bacteria turn fluorescent as you do this measurement. Right, so this means that we can perform electrophoresis without lysing water, and this means that we have the conditions on the surface to hybridize RNA to the surface. All right, so then the next question is, how much of the RNA can we actually get out of the tissue and onto the surface? To measure this, we used regular single molecule fish as the reference or gold standard. So that's shown here on the left. And we measured by counting spots, the total number of transcripts per square millimeter under optimal conditions for fish. And we think that fish, like single molecule fish, is really near 100% efficient. So this is really more or less showing all the RNA that's there in the tissue. Now, if we use our full eel conditions, which is we use electricity and we use poly-D-lysine on the surface, this is a positively charged molecule that attracts the RNA, so it helps capture the RNA. Then we get about 19% of the RNA transferred onto the surface as measured by fish on the eel slide. So 19% was the estimate here. If we don't use electricity, but we still use polylysine, we actually get this similar capture efficiency, maybe even more. But as you'll see in a minute, then the molecules are much more dispersed. So we lose cellularity, we lose the cellular resolution. If we omit the poly-D-lysine, we get very low capture efficiency. And if we omit both, then we get basically no capture at all. So we need both electricity and we need the poly D lysine to capture RNA efficiently and with cellular resolution. Right. So next question then is, what is the lateral diffusion? So one of the points of using electricity here is to speed up the transfer of RNA to the surface. You might just put the tissue on a slide and wait. And of course, eventually the RNA will diffuse out of the tissue and deposit on the slide. There's nowhere else for it to go. Well, maybe half of it will go out in the buffer, but in essence, the RNA will transfer. And this is the basis for Visium or these sort of spatial methods where you put the tissue slide on kind of a microarray, barcoded microarray, and it's just based on diffusion. But so the point of electricity is to speed up this process so that you get a directional transfer of the RNA onto the slide surface in an amount of time that doesn't allow for very much lateral diffusion. So the hope then is that this will minimize lateral diffusion and you will maintain a faithful representation of the cellularity of the tissue. So here we try to estimate this, and on the left you see RNA fish again, single molecule fish for 3 genes, MALAT1, actin, and tubulin A1A. And yeah, here you can see the cells. MALAT1 is mainly nuclear, and actin and tubulin are mainly cytoplasmic, and you can see the beautiful cellular structure. This is kind of the gold standard. With Eel, you can see that most of the, or a lot of the cellular structure is maintained, but the sensitivity is much lower, and I will come back to that in a minute. But if you compare with Eel, same conditions, but not using electrophoresis, you can see how even the nuclear MALAT1 transcript is now dispersed all over, and you basically lose the cellular structure. A different way of asking what is the lateral diffusion is shown in the bottom half of the slide here. Here we identified relatively rare interneurons in the cortex, which are VIP positive. So VIP is expressed at a very high level, but in very few neurons. So they're very sparse and distinct, and so you can quite easily identify them. And then you can ask, where are the VIP transcripts relative to this, sparse individual cells? And if you do this with single molecule fish, you get the upper picture here. So you can see these VIP molecules sort of located in the cytoplasm of the cells. If you do the same with Eel, you can see that there is some dispersion, but by and large, the VIP molecules remain relatively close to their centroid. Again, the sensitivity is lower, and I'll get back to this in a minute. You can quantify this and ask what is the distribution of VIP molecules, VIP mRNA molecules, relative to the centroid. And on the left, you can see the distribution for single molecule fish in tissue. So this is regular, this is where the transcripts are basically in the tissue. The half max is about 4.4 micron. And in green on the right-hand side, you see the distribution after transferring the RNA out of the tissue onto the surface. And you can see that it has spread a little bit, and the half max now is 7.5 micron. But still, this is sufficient to maintain, to a large extent, the cellularity and cellular resolution of this tissue blot. Right. So that's a few highlights of actual method development, and so now let me turn to showing you what the data actually looks like. So here's an example of a full sagittal section of the mouse brain. This was acquired in 2 days. It's a 440 gene experiment. We identified almost 9 million RNA molecules here. In this particular image that I'm showing you here, I'm only showing you 30 selected genes, because otherwise it's very hard to see the structure of the brain with so many genes. And I'm going to zoom in on the regions here depicted in as C and D. So just to orient yourself, you have the olfactory bulb here on the left, you have the cerebellum on the right, you have the cortex on top. You can see the layers in the cortex, you can see the striatum in yellow below, the thalamus in red, and so on and so forth. Now, if we zoom in, now this is the region that was labeled C. Now you're seeing the ventricle here. There's a lot of choroid plexus in this ventricle. You see the top part of the striatum here. And this is the rostral migratory stream. In D, you see the cerebellum. So now you have, for example, in kind of gold here, you have the Purkinje neurons, which are very characteristic, sit in sort of a line like this in cerebellum. If we zoom in further on this rectangle here, you start to now get down to single cell level. So you start to see the individual cells. If we zoom in even more, you start to see now how the mRNA molecules are located relative to nuclei. So the nuclear information here is from a DAPI stain that was acquired in parallel. And same for the cerebellum here on the right. Now you see even more of the Purkinje neurons here, one after the other, like pearls on a necklace. And the other layers here are the white matter in green, the granular cells in blue, and the molecular layer mostly here populated by Bergman glia in kind of orange brown. So this shows that with EEL, you can go from a complete overview of the brain and zoom in to any cell in the brain in basically a single experiment. Right, so using this experiment, one of the things that we were interested in was to understand the spatial regional distribution of cells and genes. So you can take the single cell or the spatial data, and you can basically cluster this in a way that is aware of the spatial structure of the data and neighborhoods and so on, and come up with this kind of regionalization map. So this shows basically regions of the brain that have similar gene expression structure. So now you see the layers of the cortex, and you see the distinction between different parts of the cortex. You see the cerebellar structure, and again you see the striatum and the thalamus, and here's the hippocampus and so on. But if you do this, you quickly realize that these borders are not all made the same. So some borders really are very sharp, and they separate very distinct regions, whereas other borders are not really sharp borders, but they're kind of part of a gradient of gene expression or a gradient of cellular composition. So to capture this, we came up with we used a sort of a local score for each voxel, so each spatial location, computing the correlation with other voxels, neighboring voxels in different directions, and to compute a border strength. So where voxels are very similar to their neighbors, there will be a very low score for this border strength. But where a voxel is very different from its neighbors, then this border strength would be high. And it's also a directional. So you actually come up with sort of not just a border strength, but a direction for the border, like here shown for the hippocampus. Now this shows, for example, that in the cortex, we can still see the layers, and they're separated by borders, but these borders are relatively weak. Whereas the border between cortex and the underlying white matter, for example, is a very strong border. And same in striatum, there are, as you saw on the left here, there are definitely different regions in the striatum, but these are not separated by strong borders. There basically aren't any strong borders in striatum. So these are more like gradients of gene expression. So as an example, if we look here through the cortex, so now we're looking at this little red rectangle here through the cortex. Here, going from the pia on the left to the white matter on the right, there is there are multiple layers that are distinguished by specific expression of key marker genes, as shown here. And the border strength metric that we came up with has a number of peaks that really do separate the layers. So you have a peak here between 2, 3, and 4, between 4 and 5, between 5 and 6A, and so on. But again, these are not as sharp borders as, for example, between the cortex and the underlying white matter. So this is the border between 6b and the white matter. You see, it's a very strong border. And in contrast, then, the opposite is true in the striatum. So here are the clustering results of striatum. Here you see there's a number of spatial regions that show distinct gene expression, and they're separated by clustering. But of course, clustering just imposes a structure on the data that looks like very sharp regions, whereas in reality, it's much more it's much more of a gradient, a spatial gradient across the striatum. So if you look at certain marker genes here, for example, you can see that they are expressed, sort of preferentially in parts of the striatum, but they really form these gradients. And as an alternative way of depicting this, instead of these sharp cluster borders, we can determine for each voxel the probability that it belongs to one of these clusters. So this is kind of a classification task, or a probabilistic classification task. And now you can depict this as more faithfully as a gradient of spatial gene expression that I think reflects much more the underlying architecture of the striatum. And now applying this to the whole brain, on the left you see same picture as I showed in the first slide. By now, instead of having these sharp cluster borders, each voxel is recalculated to show a mixture of colors that depict the probability that this voxel belongs to one of the spatial clusters from the original clustering. And again, you see the striatum has this sort of gradient-like structure. The ventricle is clearly distinct. You can distinguish the CA1 from the CA3 fields and the dentate gyrus in the hippocampus. You somewhat see the layers of the cortex, and definitely you see the anterior and posterior regions of the cortex, and so on and so forth. On the right is a different way of depicting this, where you overlay this mixed region colors with a height map that shows the border strength. So now, for example, between the cortex and the underlying white matter, there are these very sharp borders that clearly distinguish, whereas within the striatum, you don't have any sharp borders at all, only outside the striatum and towards its neighbors. Anyway, so this was all analyzed directly on the spatial data. But of course, ideally, we want to convert the spatial data into single cell data. We want to know which molecule belongs to which cell. So as a first step towards that goal, we performed segmentation of the data set. And the way we did this was to image the nuclei. So this is, I think it's a propylium iodide stain that gives us the nuclei. And we have fiducial marks that consist of fluorescent beads that are sort of sprinkled under the tissue, and that are imaged along with the nuclei and also along with the mRNA in subsequent stages of the experiment. So we have an initial image data set acquired at 40x magnification with beads and nuclei. And then we performed the Eel experiment after removing the tissue. So now the nuclei are gone, and we just had the RNA on the slide. We imaged the RNA signal, and we imaged the same beads that were imaged before, and now it's a 60x image. So we end up with a registration problem where we have to find a transformation between 40x and 60x of these clouds of beads. And once we solve this registration problem, then we can transform the 60x signal down to the 40x signal and register it to the nuclei. Then we use cell pose to find the nuclei and segment them. And finally, that allows us to assign mRNA molecules to cells or to nuclei and their neighborhoods. So this is shown here on the bottom left and bottom right. It works reasonably well. It's definitely far from perfect, and it's still quite difficult to know in many cases. And that is true, by the way, also of regular SM fish in tissue. it's not very easy to understand or to know with high level of confidence to which cell each molecule belongs. But anyway, this allows us now to generate a single cell data set from our spatial data set. And in B here, you see a t-SNE plot of now the cellular, basically the matrix of cells by genes, the gene expression matrix of the whole sagittal section of the brain. And so we clustered this and we can project it back to the section. And now you're seeing in C here, you're seeing the same section, but now it's colored by cell identity instead of by gene expression. In this segmentation procedure, we captured about 70% of the mRNA, so a bit more than 6 million molecules were assigned to a cell, and we detected about a bit more than 100,000 single cells. So that's pretty good for a single experiment that takes 2 days to perform. And if you zoom in on this map, you can see, for example, here, now we're, again, this is the striatum and this is the ventricle. You see the choroid plexus here in the middle, and you see the rostral migratory stream on the surface of the striatum. In the middle, you see the hippocampus, the C1, C3, and the dentatorus. And on the right, you see the cerebellum. At the bottom here, I'm showing a couple of cell types that we know and that have very little spatial heterogeneity. So microglia, which are dispersed all over, macrophages, endothelial cells, pericytes. And you can see how they are basically more or less evenly dispersed. And in contrast, on the right, bottom right here, you see the major classes of astrocytes in this data set. The telencephalic astrocytes, which are really confined to the telencephalon. And there's this beautiful demarcation line between the telencephalon and the diencephalon and the rest of the brain. So you have the telencephalic astrocytes marked by MFG8, the non-telencephalic ones marked by AGT. And then on the surfaces of the brain and in the white matter, you have the fibrous astrocytes marked by GFAP. And interestingly, what I had never seen before, they cover the whole brain and they cover the white matter like this, but for some reason they don't cover the cerebellum. So the cerebellum is devoid of these fibrous astrocytes. And in the cerebellum, of course, you have the Bergman glia, which is also kind of astrocyte sitting in this next to the Purkinje cells. All right, but that was a single sagittal section. But I told you this is a robust method. We can do it over and over again. So we applied it here to make a complete atlas of the mouse brain. So this is an atlas of 7 sagittal sections, going from the midline to the sort of lateral side. And here I'm just showing some examples of genes from this atlas. So it's all, of course, a single brain. It's a single experiment, and I'm just showing you subsets of genes in each column here. So, for example, in the first column here, you see the striatum in yellow, the thalamus in green, and in the second column, you see again this MFG8 astrocytes versus the AGT astrocytes, so the telencephalic and the non-telencephalic. And now you can see that it is true that there are 2 very distinct populations of astrocytes, one in telencephalon and one in the rest of the brain. And this border is super sharp and it really demarcates the telencephalon from the rest of the brain. In the middle here, showing various cell types of the meninges and the ventricles. For example, Decorin labels the fibroblasts of the meninges. And FOXJ1 labels ependymal cells along the ventricles, and so on. You can see the layers of the cortex most clearly here in the lateral sections. So you see this beautiful layering of the cortex marked by these genes. You see the hippocampus, its various fields, and the cerebellum, and so on. So it's really a really powerful method to go from a whole brain and down to single cell level with hundreds of genes. Now, one thing you can do with a spatial atlas like this is now to take cell types defined by single cell RNA sequencing from other experiments. And we had done this before. So a few years back, we published an atlas of or a census of the whole mouse nervous system So that's depicted on the top here. This is a dendrogram, all the cell types or all the neuronal types of the CNS that we defined, that we discovered in this work. But this was, of course, without any spatial information. It was just purely, abstract cell types defined by single-cell RNA sequencing. And at the time, we used gene expression profile of each of these cell types and compared it to in situ hybridization expression profiles from the Allen Brain Atlas. And you can make this pretty nice, actually, mappings of, for example, here are glutamatergic neurons of the cortex. So we have a layer 6B neuron here, and here's one that is specific to the barrel cortex, and so on. Here are the dopaminergic neurons of the midbrain in the substantia nigra VTA, and so on. But as you can see, of course, the spatial resolution of this atlas is not very high. Not least because the Allen Brain Atlas is a composite of hundreds of brains. It's not a single brain, so you have to adjust a bit for this when you do this spatial analysis. So now that we have a true single cell and single molecule fish mouse atlas, we could do this same registration again with our new atlas. So we made use of an algorithm called Tangram that was developed by Aviv Regev's group, which is a more formalized way and I think a very effective way of registering single cell data with spatial data by accounting for the abundance of different cell types and the space that they occupy spatially. We had to make a small adjustment to this algorithm because the algorithm, as it was originally conceived, requires computing a projection matrix that is the product of number of cells and number of voxels, and we have hundreds of thousands of cells and hundreds of thousands of voxels, so we quickly run out of memory. But the solution is to map clusters instead of cells, so basically the single cell data is condensed into clusters, i.e. cell types, and this is mapped to the spatial, to the voxels. This is now integrated in Tangram, and it's one of the features of Tangram that you can use. All right, so that allows us now to take the cell types that were defined in our previous work from single cell RNA sequencing. So these are various glutamatergic cells of the cortex, and map them to the Eel mouse atlas. And you can see you get this very nice spatial distributions that clearly map to distinct layers of the cortex. So here we have layer 5, here's layer 6, and here's layer 6B, and so on. And you get this from the midline to the lateral sections. Having done that, now you can also transfer labels from your single cell data to the spatial data. So one thing we had done very carefully in our single cell analysis was we had inferred the neurotransmitter phenotype of every cell. So we knew from every cell whether it was dopaminergic or glutamatergic, and etc. And this was based on genes that are involved in neurotransmission of each of those neurotransmitters. So for example, the enzymes that make the neurotransmitter, or the transporter that packs the neurotransmitter into synaptic vesicles, or the reuptake transporter that clears it out of the synaptic cleft. So that was our definition. And of course, in the single cell data, we had all those genes measured, so we could infer that for every cell. In our spatial data, we didn't have all those genes measured. So we couldn't directly determine the neurotransmitter phenotype of every spatial location. But once you have mapped all the cell types of the brain from single cell data onto the spatial coordinate system, now you know in each voxel what is the composition in that voxel of cell types. So you know that in this voxel there is 80% of this cell type and 20% of this other cell type. And so now you can project information from the single cell data onto the spatial data. And here's an example where we projected the neurotransmitter phenotype of cells onto the spatial map. And now you see, for example, in the cortex, there is clearly a mixture of glutamatergic, specifically VGlut1 cells, and GABAergic cells. So there is a kind of a red-blue mix here. And there's more of the interneurons in the superficial layers and more of the excitative neurons in the deeper layers. You also see that the striatum is really very dominated by GABAergic neurons, obviously. You see that the thalamus is rare in having a combination of both VGLUT1 and VGLUT2 glutamatergic neurons. And you see the beautiful layered structure of the olfactory bulb here that has really all the different neurotransmitters in beautiful layers like this. Yeah, and so on and so forth. Down here in the midbrain, you see the dopaminergic neurons of the midbrain, for example. All right. Last couple of slides. So all of this is mouse, obviously. One of the reasons we developed EEL was that we were hoping that it would solve one of our biggest problems with our previous methods, which was that we couldn't really apply them to human brain. And the reason was that in human brain, unlike in mouse brain, there is this dreadful autofluorescent precipitate in the brain. So if you look at the brain section from humans, it's full of these highly autofluorescent speckles called lipofuscin. And they are autofluorescent in all channels. They're very highly autofluorescent, and they basically destroy all hope of doing single molecule fish in human brain. So our hope was that now we're transferring the RNA out of the section and we get rid of the tissue section, then the lipofuscin would also be gone. And that turns out to be actually almost true, not perfectly, but to a very high extent. So on the left here, you see a tissue section before the EEL procedure. And then on the right, you see the same tissue section after the EEL procedure. And now there is still some remaining lipofuscin, these bright dots. But now you start to see these very faint dots, which are zoomed in here. These are the actual RNA molecules that this is the EEL signal. This is the fish signal that we're looking for. Each dot here is a single RNA molecule that we decode and identify. So on the left-hand side, they are still there, but there's no way to see them because of the extremely bright autofluorescent signal. So this means that happily now we can apply EEL to human brain. And so here's an example that was run by Alejandro in my lab in collaboration with Adeline and Rebecca Hodge and Darkin from Allen Institute, where what you're seeing here on the left is about a square centimeter of the human visual cortex. It has this characteristic blue band here below layer 4, and here we're measuring now 448 genes. This on the left, we're showing all the genes, so it's hard to distinguish anything. We zoom in, we start to see the cellular structure of this tissue. So this is a zoom in of this square here. But it does show that we are able to run now EEL on human visual cortex. If we flip it over, same tissue section, now it's flipped over just to make it larger on your screen. Here we're showing from the same data set in each of these different regions, we're showing a different subset of the genes to make clear different structures. So for example, at the top here, we're showing GFAP in magenta. So these are astrocytes of the glia limitans. And we're showing PLP1 in yellow. So these are likely oligodendrocytes. If you go to field 4 here, this shows various interneurons. We have GAD1 and GAD2 in blue, NPY in green. And I think you can see a single NPY cell here in green. I don't know if you can see it on your screen, but I hope so. Zooming in here, this square here is zoomed in here. Unfortunately, it's also rotated 90 degrees, but here you see this green cell again. So this is a single NPY positive interneuron. So again, showing you can go from the large to the small. On the top here, you're seeing our, this is a neural network-based inference of spatially defined cell types. And here you see astrocytes, for example. You see somatostatin interneurons. You see reelin positive neurons in the layer 1. You see various excitatory neurons of layers 2, 3, 4, 5, and 6. And then you see the oligodendrocytes of the white matter. All right, finally, we also started applying this to developing human brain. So here is an example of an atlas that we're working on. In this experiment, we performed the 448-plex eel fish on a whole human head at 7 weeks post-conception. And we have here 25 sagittal sections. I'm showing you a single sagittal section here, but we have 25 sections through the whole head. So it's a really comprehensive atlas. And here I'm showing you all of the genes. So it's a bit difficult to orient yourself, but I will point to the spinal cord coming up here into the hindbrain, midbrain, and forebrain. You have the eye here somewhere. You have the mouth here and the tongue pointing towards the left. And there are so many things that you can look at in this atlas. It's a really beautiful data set. I just want to show you 2 examples just because they're pretty. The first one is the eye. So this is the developing eye in the human 7-week embryo, showing just a subset of the genes now for clarity, but you can really see very beautifully the future lens, the retina. You see the various structures of the front of the eye here going to be the cornea and so on. And really corresponds really to this histological section from and this is from Gray's Anatomy at about the same age. And of course, with the difference that now we have genes, so we can start to say something about the function of these cells. And the second example, which I think is also very pretty from the same atlas, is the developing tongue. So here's the tongue. This is the tip of the tongue. This is the sort of dorsal part of the tongue. It's defined by these genes, but really what is amazing here is this row of single cells expressing sonic hedgehog. This is the exact moment when taste buds are induced on the surface of the tongue. So these are going to develop into structures that form taste buds on your tongue eventually. All right, that's all for today. All of this data is available at mousebrain.org. Alejandro developed this beautiful viewer that you can install to browse these data sets. Not sure how smooth it is on your screen now because we're doing it over Zoom, but it's really a beautiful tool that allows you to very smoothly and nicely zoom in and out of the data set and show different genes and so on. And importantly, this scales to many tens of millions of spots, which has been very challenging with other viewers. So in summary, EEL gives you fast centimeter scale imaging. Takes 48 hours to do an experiment. We are now doing up to 1344 genes in 3 channels. I didn't show you that today. That's very much ongoing work, but we are scaling it now by just adding color channels in parallel. It's a robust routine workhorse method. Detection efficiency is about 4 or 5%, so that's a drawback, obviously, but I think it's sufficient for many kinds of analysis. And very importantly, it enables human brain RNA fish. And with that, I want to thank all the contributors. I thank the key people along the way, but I want to thank all the rest of the people in my group, collaborators and funders, and all of you for listening.
Yeah, thank you, Sten. That's kind of mind-blowing, very impressive. Already quite a few questions in the chat box, but actually, Roy, you raised your hand. So Roy, you can go ahead.
Hey, Sten, really beautiful work. One question that I had, so one of the motivation behind your original Awesomefish at the time, and one of the challenges we all have in combinatorial fish is optical crowding, right? When molecules become sort of too dense, which I imagine
Yes.
could have been even a bigger problem here if your capture efficiency was actually closer to 100%, right? Then you wouldn't be. So how far is this 4% from actually being the point that if you push it any further high, you will get to the optical crowding problem?
Yeah, you're exactly right. So I think we could maybe increase the sensitivity a bit before hitting that, but not very much. So there is really a very clearly a trade-off between the number of genes detected and the sensitivity. So if we would have, if our sensitivity were to be 100%, then we wouldn't be able to do 400 or 500 genes per channel, definitely.
Okay, Alex.
Hi, great talk. Yeah, I was just curious. Since everything is on a surface now, are you still using confocal, or can you take advantage of something like Perf or a different microscopy method?
No, none of this is confocal. It's old school epifluorescence microscopy. It's 60x objectives and oil immersion.
All right, so Emmanuel.
Thanks, very amazing. I would like just to ask you, exactly, I didn't much understood how you managed to segment the cells and how you distinguish different cells between them.
That is very much an ongoing challenge. Right now, we do it by segmenting the nuclei based on the DAPI stain, or really it's a propylium iodide, I think, but yeah, equivalent to DAPI. So we segment the nuclei and then we just expand them. So we take a particular distance, well, we expand it in the shape of the nucleus, kind of. This is clearly suboptimal because cells are not shaped like that. So one kind of remedy that we're working on right now is to apply PCI-seq, which was this probabilistic method developed by Kenneth Harris that makes use of prior knowledge about which molecules belong in which cell types, and we kind of use this in a circular expectation maximization algorithm to assign spots to cells that they most likely belong to, in a way. And so, yeah, but this is really very much an ongoing challenge for, I guess, for most of these methods. It's not easy. But also say that I think there is a completely different way of analyzing this data where you don't do the segmentation and instead analyze on the level of spots and spot neighborhoods and use neural networks. I didn't have time to go through that, but basically what I showed from the human brain was analyzed in that way, where you're looking at each individual spot and its neighbors and clustering those instead of clustering cells.
Thanks.
Yeah. I remember we had a very similar discussion last year. So whether or not in a spatial context, the segmentation is absolutely critical, or in some cases, maybe not necessary. Actually, the cells, when they are in contact, they can exchange materials, since proteins, molecule, transcripts. That has been reported. Just don't know how much impact that is. Might be ubiquitous. Yeah. It's one open question.
But also in the brain, a large proportion of the volume of each neuron is very far away from the nucleus, obviously.
Yeah.
And there, all these white matter tracts obviously belong to cells, but there is no way of assigning them to a cell.
Mm-hmm.
So, yeah.
So maybe Ben, you can go ahead.
I have a question about how you approach handling off-target binding of your fluorescent readout probes. So in your probe design, are you thinking about sequence specificity? And then the other part of that question is, do you worry about the poly-D-lysine, just sort of a gradual increase in background fluorescence with repeated staining, because maybe probes are sticking to the poly-D-lysine?
Yes, right. So for the first part, when we design the probes, we do take into account the rest of the transcriptome, and we try to omit the probes that would cross-hybridize. This might sometimes make it impossible to design enough probes for a particular gene, although in the vast majority of cases, it is still possible to design probe, because a probe, when I say a probe, this is actually a set of probes. So we tile the whole RNA molecule, right? So ideally, we want to fit at least 25 encoding probes on an RNA. We can make do with a bit less, but of course, if we can, we would like to put 40. And we avoid those regions where there are homologies.
Sure.
As for the second part of the question, so the limitation to the number of cycles that we can do is indeed the loss of signal and the increase of the background. So the signal to noise ratio. And not sure exactly what causes those things to happen, but I guess it's a variety of things, including accumulation of background due to non-specific hybridization, but also loss of RNA molecules or degradation of RNA and so on. So I should say, I didn't say it here, but we kind of use multitude of approaches to really stick our RNA to the surface. So we have an oligo-T probe or oligo to bind the poly A tail. Then we have the poly-D lysine, which is electrostatic binding, but then we also cross-link. So we have a fixation step to really make it stick.
Very cool. Thank you.
Yeah, thanks, Ben. Salman?
Simeon, yeah. Hi, so 2 questions. Great talk. First of all, thank you. Yeah, first is kind of stupid, maybe. I actually didn't quite understand how the RNA gets polarized so that it reacts to the voltage. Maybe I got something wrong, not sure there. But and second question was for you mentioned Kenneth Harris's PCI-seq. So does that operate on the level of kind of fuzzy sort of like expansion, or on the level of assigning or classifying after expansion?
Yeah, so the first question, RNA at the pH, at physiological pH, RNA is negatively charged. So every nucleotide has a negative charge. It's a phosphate. The phosphate backbone has a negative charge. So it's the same with DNA. That's why electrophysics works.
Yeah.
But that's also why it wouldn't work if the pH would drop, because then it would lose its charge and it would no longer work.
Okay.
Right, but and what was the second was the PCI-seq. This is a purely computational method. It's been used so far in conjunction with specifically in situ sequencing, but there is nothing to prevent it. It's a general method that can work with any kind of spatial, and it basically gives a probabilistic assignment of RNA molecules to cells. So you give the algorithm a spatial distribution of cells. So basically, for example, nuclei or whatever your segmentation algorithm gives you, and you give it all the spots. And then you give it reference data from single cell RNA sequencing. And so it knows, for example, if a cell has GAD1, which is an interneuron marker, it's also likely to have GAD2. So the algorithm then kind of tends to assign GAD2 molecules in the vicinity to that same cell. And it's probabilistic, and in the end, you end up with probabilistic cell type calls. So each cell in your data set will be 80%, somatostatin interneuron and 20% NPY interneuron or whatever.
And that.
Okay.
So thank you. Just a super quick follow up on that. So does that carry on through in the so basically it's kind of like assigning pixels, right? With this method, because the genes are sort of can be seen at, they have like a single location, right, on the on your image.
Right.
And so this kind of, I call it fuzzy, but you can call it like a distribution of assignments, right? Because it's like 60% this, 20% this.
Exactly.
So then that carries on through in the analysis, and you kind of take that cell as a type according to that distribution. So.
Yeah, right. And I mean, you keep the probability distribution. So if you want, then you could, for example, extract only the high confidence NPY interneurons and look at those. That gives you a more fuzzy representation that can be useful for downstream analysis.
Thank you.
Right. Thanks. So in the interest of time, I think Nachiki, I will give you the chance to ask the last question, and then I will quickly go over the question in the chat box. I really think most of the questions have already been answered, but I will see if any remaining questions. Sten, if you don't mind, I will quickly go over those questions with you after Nachiki.
Yep.
All right.
Thanks,
Nachiki, go ahead.
Yep. Thanks, Rong. So great talk, Sten. So a couple of questions. So number one, in what state the tissue is? So is it like cross sections, FFP material, or what sort of tissue that is?
It's fresh frozen cryosections.
Right. And have you tested on FFP? I mean, is there a way forward to do that? And okay, not really.
I don't think so, because the RNA will be degraded and it will not
Right.
transfer intact. It will transfer in pieces. I don't think that will work.
Right.
I can be wrong, but yeah, we haven't tried.
Okay. And the second question is, I mean, you showed some fantastic data when it came to, let's say, the brain tissue. So how versatile is the approach when it comes to doing different types of tissues, different species, and so on?
Yeah, we have only applied it to brain. We've applied it to adult human, to embryonic human, to adult mouse, and also to glioblastoma. So those are the ones we have experience with. I don't think there is any particular reason to why it wouldn't work with other tissues, but you might have to optimize the digestion step.
Sure.
This is kind of a step that has to be adjusted a bit so that you don't over digest, but also, yeah, you have to, it depends on how tough the tissue is, basically.
Yeah, thank you.
Yeah, thanks. So now I will quickly go over the question in the chat box. I think most have been answered actually. So one is the RNA quality. So you see you capture actually lower efficient compared to without electrical. So that in part due to the RNA degradation by the electrical force apply, or that's not the case.
I don't know. So to be honest, we still don't really understand how the RNA is captured. And this is why we have 3 different ways of capturing it, because we couldn't figure out yet exactly what is the mechanism there.
Mm-hmm.
So I think it's more likely that there is something there that, I mean, it's a very complex system. You have the RNA molecules coming down and you kind of have to permeabilize the tissues to allow the RNA to move. But if you do that too much, then the RNA will just disperse into the buffer.
Yeah.
So you have to do it just right, and then it has to move down. And then you have the paradox of, in order to do the electrophoresis, you really shouldn't have any buffer. You should not have ions in there because that will counteract the electrophoresis. So the less ions you have, the better. But for hybridization, you need more ions. You need salt, and you need to promote hybridization using salt. So it's kind of, there are many contradictory phenomena going on here.
Mm-hmm.
So yeah, we don't understand it, and we, or I should say, Lars and Simone spent so much time trying hundreds of different conditions, and it's to some extent this what we have now is a result of empirical testing, but not really understanding.
Mm-hmm. Yeah. So next, can you also infer the cell morphology since you are seeing this kind of spatially defined spots?
Sorry, what is the question?
Can you also infer the cell morphology in your data?
The cell morphology. I mean, they will be fuzzy because of I mean, there is lateral diffusion, and the lateral diffusion is several microns. And so, the cell morphology will be fuzzy, but we haven't looked at it, but I think we can sometimes we can recognize shapes of cells that are, or classes of cells that have distinct shapes, but I'm not sure to what extent that is possible.
Okay, so
I mean, the low sensitivity also means that it's not as, when you have a single molecule fish experiment, you really you see the whole cytoplasm full of spots. And that gives you a lot of structure. We don't see that really in the same sense.
Okay. Yeah, actually, that's kind of, I take advantage of my role here to ask probably the last question. So I really think your technology has very unique advantage, which is the tissue is still there, right? So after you capture, so can you do something like a standard nasal staining to get a morphology, or you do multiplex immunofluorescence, and you really combine multiple modalities to further enhance your data?
In principle, probably yes. I mean, we do the nuclear staining, so that's definitely possible, and we do that before we remove the tissue. So there's nothing in principle to prevent you from doing an immunofluorescence assay or some dye staining.
Have you tried NISO? Because I'm part of the Brain Initiative. I think all other teams are doing NISO. So if we want to integrate data, you haven't. Okay.
I mean, in Visium or in the what was previously called spatial transcriptomics, it's very similar. You have a tissue and you're blotting the RNA onto in this case, a microarray. And in those methods, they always routinely perform an H&E stain. I should say that sometimes also preclude, like the H&E stain is not easy. It sometimes can make the tissue. It can partly destroy the tissue a bit. So it's but in principle, it should be possible to perform a histological stain first and then the RNA transfer.
Or after RNA transfer, you can still do
I mean, after the RNA transfer, we digest and then the tissue is gone.
Okay.
So you have to do it before.
Okay, I see. All right.
It's kind of done as part of a single protocol. So at least the way it works now, I don't think you can do it after.
Yeah, but at least you can do that from adjacent sections. All right, so thank you so much. This is super exciting technology and truly mind-blowing. And let's give the speaker another round of applause.
Thanks for having me.
I'm gonna do the real one.
Thanks.
Yeah, thank you, Sten, and thank you everyone for attending. And hope to see you guys next week at the same time, 2 p.m. on Friday. Yeah, thank you. Have a good weekend.
Bye.